Hello and welcome to BioRules video lessons. Today we are going to be talking about proteins and how they get influenced by the environment around them. In extreme situations, protein may get denaturation. It means get totally different from what's supposed to be, and that's kaput, that's it. Okay. So, proteins, they have a certain shape. They have a three-dimensional shape that's made by the interaction between the radicals that we saw in previous class before. And if you have small changes in the conditions of the surroundings, in the solution where the protein is, for example, that may lead to small changes in the protein conformation. Because if the temperature changes a little bit, hydrogen bonds may be made or may be broken. If the pH changes, hydrogen bonds may be made, may be broken, or ionic bonds may be made or may be broken. So depending on the conditions, then the three-dimensional conformation of the proteins may change as well. So as I told you before, temperature and pH are very important factors affecting the three-dimensional shape of proteins. Okay? So they have a very big influence, especially because of hydrogen bonds, sometimes because of the ionic bonds. Very good. Just to illustrate the idea, this here is a diagram of what would be a protein. So if you warm the solution, what may happen is that the hydrogen bonds may get a little bit loose and the protein may get a little bit more relaxed, with a little bit more slack, a little bit different shape. So it might not be a big deal. The protein may still kind of work, but it's not exactly the same anymore. The same happens if it gets too cold. Some hydrogen bonds may be broken, some hydrogen bonds may be made, and the protein might change its configuration. As you can see here, it is still more or less the same protein, but not exactly the same. There was a slight change in the three-dimensional conformation of the protein. Okay. There is an animation here that we are going to be seeing in class that can be very helpful to illustrate this when we cook an egg. Cooking egg is changing the three-dimensional shape of the egg white protein, but you'll see this later. Now I want to show you here what happens with temperature and the proteins, okay? So they have the natural shape, they have the three-dimensional shape where they work better, and that has evolved to be that shape in a certain temperature. If the conditions get colder or warmer than the conditions where that proteins have evolved to be, then the protein gets different. It will have a different shape, and with a different shape it may have a different function. It may be a slightly different function, it may be a totally different function. So that's something that may happen. Here an example, a hot spring in Yellowstone where temperatures can get above 90 degrees Celsius on the surface, and the bacteria that live in there, they have evolved to that temperature. So 90 degrees Celsius, they say it's okay. But if you push the temperature even higher, if you make the temperature be colder, then the proteins are not going to be working as well, and then those bacteria are not going to have an easy life living there. The same, for example, for this fish that live just under the ice. These fish have evolved to live in temperatures around 5 to 10 degrees Celsius. So they like 5 to 10 degrees Celsius. 5 to 10 degrees Celsius for them is very good. But if you increase the temperature a little bit, change the proteins a little bit. If you change a lot, then it changes a lot the function of the protein. So all this can happen because we've changed the temperature. And an example of changing temperature that's extreme is cooking. So when we cook, we heat up our foods. So we really change the temperature. So when we change the temperature, we change the proteins. So you know the example, I bet you all have cooked an egg before and saw that the quite transparent egg white becomes white, and that is changing the protein. So when we cook, we do that to kill the pathogens, because the proteins of the pathogens get destroyed and they don't manage to survive as well. But we also cook to change the texture of the food. Usually food gets softer when we cook. Another example, the pH we mentioned before, and we can apply pH to food. So If we apply different pH to a protein, like adding vinegar to a food, we are also going to be changing the shape of the protein, sometimes even drastically. And we do that in order to kill pathogens, to soften the food, and sometimes even to cook. I don't know if you had experience, but there are some meals that are made by adding acid, usually lemon, to meat, to fish meat usually, and they make ceviche. So extreme pHs are going to make extreme difference in the protein shape as well. Okay, this brings us to the idea of optimal. I said that the protein may be slightly changed or changed a lot. So what is the shape that the protein should have to be doing its best work? Well, that's going to be when you have the protein in its optimum. And to illustrate the optimum in relation to temperature, proteins have an optimum temperature to work. Ours, for example, they work very well around 37 degrees Celsius. So here, there's a typical curve of how temperature influences protein function. Usually very cold, the protein doesn't work very well, and then you go increasing the temperature, it increases slowly, so it goes increasing the functioning of the protein until it gets to the optimum, to the maximum, that temperature in which the temperature works at its best. In case of temperature above that temperature, the protein changes and keeps changing, and now changing for not so good anymore. And then it can make a drastic change until it stops working at all. So the one at the best is going to be 37. In our case, 37. In the case of the bacteria in the hot springs in Yellowstone, 90 degrees Celsius. In the case of the fish under the ice, something between 5 and 10 degrees Celsius. That's going to be the optimal. The optimal is the temperature that the protein have evolved to be, and it works at its best. What about the optimum pH? Okay, you can also have an optimum pH. Proteins work better at a certain pH. So in this example here, the protein has an optimum pH, and if you move away from that optimum pH, the protein doesn't work as well anymore. For example, proteins that work in our mouth, like salivary amylase, they work very well at neutral pH, around 7. If you make the pH become a little bit more acidic, like 6 or 5, the protein is not going to be working so well anymore. And if it makes it a little bit more alkali, like 8 or 9, it's also not going to be working so well anymore. But other proteins, they have other optimums. For example, the enzymes in our stomach, they have an optimum pH around 2 or 3. So if you put it in pH 7, it's not a very good pH for that. The optimum will be around 2 or 3 in our mouth. So every protein has an optimal pH. Okay. Okay, I mentioned some change in the functioning, and some of these changes, they may be reversible. So I will show this again with the temperature. No, small changes may be reversible, big changes might be irreversible. So let's take a look on this using an example on temperature. This here is a graph that shows the optimum temperatures, that peak over there. And if you change temperature a little bit around that optimum, the function of the protein changes as well, but changes slightly, and usually changes and is reversible. Your body, if the temperature goes to 35, the proteins are not going to be working so well, but if you warm up your body, they will work back and you're not going to be in big problem. And the same, if your temperature is 37 and your body temperature goes to 38, 39, they might not be working so well, but if you put it back to 38, 37, then the protein is going to be working back again. So this is reversible around a certain range, a small range around the optimum. What happens then? Then it's reversible. What happens if you cool down a protein? Cooling down is interesting because the protein will work less and less and less for two reasons. One, the shape of the protein gets changed because of the hydrogen bonds, all the stuff that we saw before, and also because the particles are not going to be moving so much, because the colder it gets, less movement of the particles you have, so they probably are not going to be working so well anymore. The good news are that cold temperature usually is reversible. If you cool down a protein and then you warm it up, the function go back again. If you get a human protein and you go from 37 to 5 degrees Celsius, it's going to be almost stop working. But when you warm it up from 5 degrees Celsius to 37 degrees Celsius again, that protein is probably going to be working. So why do we die if you get hypothermia? Oh, because that's a different story. And I'll tell you about that in class if you remind me to tell this story, okay? But the proteins usually, the function is reversible in cold temperature. Exception goes sometimes for freezing. When you freeze, sometimes stuff with water don't like freezing, and then it might be a big problem. What about hot temperatures? Ooh, extreme hot temperatures, then they change the protein so much that the change is not reversible anymore. And this is basically the idea of cooking. You can cook an egg, but you cannot uncook it if you cool it down. But if you cool down an egg, if you put an egg on the refrigerator and then you warm it up, oh, it's an egg white again, it's not a big deal, unless you freeze it. If you freeze, then you have a problem. Okay? So, very hot temperature, usually irreversible change. What about pH? Does pH have this problem with reversibility? Yes, it does. There is an optimum, and around the optimum, usually, this depends on the protein, but we'll say that around 1 pH, so if the optimum is 7, something up to 6 usually is not a big deal, something up to 8 is not a big deal, so around the optimum, things are reversible. But if you move very much away from the optimum, then the change gets irreversible. The protein changed so much that it cannot change back the way it was. So it gets totally reversible. And this irreversibility is called denaturation. The protein gets unnatural. The protein gets in a shape that is totally unnatural, not working anymore, and not moving back to what it was before. So this is the idea of denaturation. Irreversible change to the protein shape and the protein function, which, by the way, is what we do when we cook. Cooking is denaturating proteins, okay? So there is irreversible change in the protein shape and function, usually with extreme conditions like extreme temperatures or pH, and you have total loss of function. I hope you have enjoyed this class. It was a lot to get. There's a quiz here for you to check out your understanding, and I'll see you in the next class. Bye-bye.